Welcome everyone. Let us see the continuity of chapter 1, asexual and sexual reproduction in plants, under which we will be seeing vegetative reproduction, that too by artificial methods. In the last videos, we saw about the natural methods of vegetative reproduction. Apart from this natural methods of vegetative reproduction, a number of methods are also used in agriculture and horticulture to propagate plants from their parts. So such methods are called as artificial propagation. So some of the artificial propagation methods have been used by man for a long time and are called as conventional methods. So man used certain methods for a very long time. That method is known as conventional methods. Nowadays, technology is being used for the propagation to produce large number of plants in shorter period of time. So such methods are called as modern methods. So we can broadly divide this artificial methods into two types. One is conventional methods and modern methods. So let's start with conventional methods. So there are three common methods of conventional propagation, namely cutting, grafting as well as layering. So first one is happen to be cutting. So as the name indicates, cutting, it's a method of producing a new plant by cutting the plant parts such as root, stem and leaf from their parent plant. So from the parent plant, any part of the plant body like root, stem or leaf may be cut and can be grown. Such a method is known as cutting. The cut part is placed in the suitable medium for the growth. It produces root and grows into a new plant. So depending upon the part used, it is called as root cutting, as in case of malus. So this is nothing but the malus plant, whereby the root part may be cut and can be grown into a new plant. And stem cutting, as in case of hibiscus, bougainvillea and moringa. So hibiscus is shown here. So the stem may be cut and from the stem part the young plant can be grown. And leaf cutting, as well as in case of begonia and bryophyllum. So it is nothing but the leaf region where it develops into a new plant. The begonia plant is shown in this picture. And stem cutting is widely used for propagation. So among which the stem is used for mostly in these three cases. So the next type is happen to be grafting. So in case of grafting, the parts of the two different plants are joined so that they continue to grow as one plant. So two plants are essential. They are combined together and to grow as single plant. Such a method is called as grafting. Of the two plants, the plant which is in contact with the soil is called as a stalk. So the plant which is in contact with the soil, so this is known as a stalk. And the plant which is used for grafting is called as a scion. So the plant which is used to be as a grafting is known as the scion. Examples: citrus, mango and apple are used in case of grafting. So there are different types of grafting based on the method of uniting the scion and the stalk. So they are bud grafting, approach grafting, tongue grafting, crown grafting and wedge grafting. So this question you may get in 2-mark point of view. What are the different types of grafting methods? So bud grafting, approach grafting, tongue grafting, crown grafting and wedge grafting are the different methods of grafting. So the bud grafting, a T-shaped incision is made in the stalk and the bark is lifted. So this is nothing but the stalk or nothing but the stalk part of the plant, and a T-shaped incision is made and the bark is removed in this manner. So the scion bud with the little wood is placed in the incision. So the scion is taken and is just inserted into the cut region of the stalk part. So the scion bud with the little wood is placed in the incision beneath the bark and properly bandaged with the tape. So finally it is just taped with the tape, bandaged with the tape. So finally it gets firm and from the region where the plant will just grow. Such a method is called as the bud grafting. The second type is approach grafting. In this method, both the scion and the stalk are remain rooted. So the stalk is also rooted and the scion is also rooted. So both are found to be having the root presence. So the stalk is grown in a part and is brought close to the scion. So here the plant body with the part is nothing but the stalk and which is brought closer to the scion. Both them should have the same thickness. Both the region of the plant should have the same thickness. Now if you could see, both the plant parts are happen to be of the same thickness. A small slice is cut from both and cut surfaces are brought near the and tied together and held by a tape. So from the stalk region, a small region is peeled, and from the scion region, a small part is peeled, and both of them are kept close together and they are firmly tied together with the bandage. After 1 to 4 weeks, the tip of the stalk and base of the scion are cut off and detached and grown into a separate plant. After 1 to 4 weeks, the plant body will be cut from this region and just they will get detached and just they are grown as in a separate pot to grow as a young plant. Such a method is called as approach grafting. The third method is tongue grafting. A scion and stalk having the same thickness. Now if you could see, this is said to be the scion and this is said to be the stalk. So both of them have the same thickness and is cut obliquely. So they are cut in a cross manner or oblique manner, and the scion is fit into the stalk and bound with the tape. So the scion is exactly kept on top of the stalk and they are bounded by a tape. Such method is known as the tongue grafting. The fourth type is called as crown grafting. When the stalk is large, but here when you see, the stalk is large whereas the scion is thin. So when the stalk is large and the size of the scion are cut into wedge shape and are inserted on the slits or clefts on the stalk and fixed in the position using the graft wax. So here the scion part or the lower part is cut in a wedge shape manner, and this scion is inserted into the stalk region, and finally they are applied by certain graft wax so that they get firm and fixed together with the stalk region, and from there the plant will germinate to come a rise. So such a method is called as crown grafting. And the last method is wedge grafting. In this method, a slit is made in the stalk and the bark is cut. So once when you see, the stalk is cut in a manner where the slit is cut in this stalk region and the bark is also cut out. A twig of scion is inserted and tightly bound so that the cambium of the two is joined together. So a little bit part of the scion is also cut and the scion is inserted into the slit portion of the stalk region, and they are firmly tightened with the bandage, and both the cambium region of this scion and the stalk will get combined together and they just grow into a new plant. Such method is called as wedge grafting. The third type is happen to be layering. In this method, the stem of a parent plant is allowed to develop roots while still intact. So when the root develops, the rooted part is cut and the plant to grow as a new plant. Here, in the case of layering, the stem part is usually buried under the soil and a certain weight is kept on top. So after some time, the stem region will get detached from the parental body and later this will develop into a young plant. So examples: Ixora, Jasminum, mound layering and air layering are the two types of layering. So under case of layering, there are two types. One is happen to be mound layering, and the second type is happen to be air layering. So the first one is happen to be mound layering, where this method is applied for plants having flexible branches. So the branch is said to be flexible. So this can be bent. So the lower branch over the leaves is bent to the ground and part of the stem is buried in the soil. So the lower part of the stem will be kept under the soil and they are buried under the soil, and the tip of the branch is exposed above the soil. So the tip of the plant will be just grown above the soil level. After roots emerge from the part of the stem buried in the soil, after some time some roots will generate from this particular region. A cut is made in the parent plant, so later a cut is made in this region and the parent plant so that the buried part grow into a new plant, so that this will generate into a new plant. Such a type is called as mound layering. The next is happen to be air layering. In this method, the stem is girdled at the nodal region. So here if you could see, the stem is girdled near the nodal region, and certain hormones are applied to this region which promotes rooting. Certain hormones are also applied in this particular region to generate the roots. So this portion is covered with a damp or moist soil using a polythene sheet. Either this is covered by certain moist soil or by using certain polythene bags or polythene sheets. Roots emerge in these branches after 2 to 4 months. So from these areas, the roots will emerge later regarding 2 to 4 months. So such branches are removed from the parent plant and grown into separate pot or ground. Later this area is cut down and they are separately potted in certain regions and where they can grow into a new young plant. Such a method is called as air layering. So advantages of conventional methods. The plants produced are genetically uniform. So the plants will be uniform genetically in case of the conventional methods. So many plants can be produced quickly by this method. So enormous number of plants can be produced at one time by the application of conventional methods. Some plants produce little or no seeds at all, whereas some seeds produce, they are incapable of germination. In such cases, the plants can be produced by applying the conventional methods. Some plants can be propagated more economically by vegetative propagation. Example, Solanum tuberosum. In case of potatoes, this can be propagated for the economic reasons enormously. And the last point is very important. So two different plants with desirable characters, such as one plant will be disease resistant, the second plant will be having the high yielding capacity. So both are grafted together and a new plant is got, so which will have the same desirable characters. For this reason, the conventional method is very important. The disadvantages of conventional methods. Use of virus infected plants as parents produce virus infected new plants. So once when we select virus infected plants, which may also produce the young ones also will be infected by the virus. So vegetative structures used for propagation are bulky and so they are difficult to handle as well as to store. So this is also one of the disadvantageous characters. The next method is modern method. Professor F.C. Stewart in the year 1932 of Cornell University showed that the mature flowing parenchyma cells removed from the carrot were placed in a suitable medium under controlled conditions could be stimulated to start dividing again to produce a new carrot plant. So from the mature flowing parenchyma cells, Stewart got an entire carrot plant. Such a method is called as totipotency, and the cells having that ability to develop into a young plant is called as totipotent. So this characteristic feature of cells is utilized in horticulture, forestry and industries to propagate plants. So the technique is widely used in the field of horticulture, forestry and in industries. So the growth of plant tissue in special culture medium under suitable controlled conditions is known as tissue culture. So this question you may get in 2-mark point of view. Define tissue culture. So micro-propagation. So the regeneration of whole plant from a single cell, tissue, or small pieces of vegetative structures, namely root, stem or leaf, through the tissue culture is called as micro-propagation. So this is one of the modern methods used to propagate plants. And advantages of modern methods. Plants with the desired characters can be multiplied rapidly, short duration. So here, this is very important point. Plants with desirable characters can be multiplied very enormously in a shorter period of time. Plants produced are genetically identical, so they are uniform in genetic manner. So tissue culture can be carried out in any season to produce plants. So there is no season. So there is no season at all for the production of this plants under tissue culture method. Plants which do not produce viable seeds and seeds that are difficult to germinate can be propagated by tissue culture. This is also very important. So the plants which are not able to produce the viable seeds and which are unable to produce seeds at all, so they can also be able to germinate by applying the tissue culture method. So rare and endangered species of plants can also be propagated. Disease-free plants can be produced by meristem culture, and cells can be genetically modified and transformed using tissue culture methods. Disadvantages of modern methods. So it is labor intensive and requires skilled workers. So it is always very important. It needs certain skilled workers to do this actual process. So sterile condition must be maintained, which adds to the cost and which is also expensive. Since the clones are genetically identical, the entire crop is susceptible to the new diseases or changes in the environmental conditions will wipe out the species. So because they are genetically uniform and same, the entire crop is affected by particular diseases and the environmental changes cannot suit them. so that the plant species will get wiped easily. Sometimes callus undergoes genetical changes which are undesirable for the commercial use. So with that, this artificial method is getting over. Regarding the other topics, we'll see in the next videos. Thank you.